Welcome to the In-Depth Academy. My name's Dr. Geraint Parry, and today I'm going to introduce the plant nuclear pore complex. So I can imagine when you think about a cell, you think of two main components, the nucleus and the cytoplasm. And we all know that the nucleic acid is within the nucleus, messenger RNA is made there, that messenger RNA travels out into the cytoplasm where translation occurs, and then proteins move wherever they need to go within the cell. But often people don't think about this section in between. So how is it that the messenger RNA gets from the nucleus out to the cytoplasm? And how is it that newly made proteins go from the cytoplasm, if needed, back into the nucleus? And this is what happens. This process goes through the nuclear pore complex. So if we look at the structure of a nucleus, we can see that the nucleic acid is in the center of the nucleus here. This is where transcription occurs. And then there is a double envelope, a double lipid envelope, which separates the nucleus, that segregates the nucleus from the cytoplasm. And then within this envelope, there are these holes called nuclear pores, and then the nuclear pore complex sits within these holes. And this is what controls the transit of molecules between cytoplasm and nucleus. So if we look at a cartoon of this occurring, that you can see that smaller molecules are able to move through the pore, whereas larger molecules are oftentimes prevented from doing it. And what's key is that this is an active process. So this doesn't just happen by chance. The nuclear pore complex is responsible for binding to different proteins, which then will allow the transport of different molecules. And you can see there are a number of different components which make up the entirety of the nuclear pore plant complex. On the nuclear plasm side, there's this nuclear basket, while these cytoplasmic filaments stretch into the cytoplasmic region. If we look in a little more detail at this region, you can see there are multiple subcomplexes within a single nuclear pore complex. As I said, the nuclear basket, the cytoplasmic filaments, and then there are more structural components which help anchor the nuclear pore complex into the nuclear pore. And then, of course, there are transport components which are actually responsible for moving molecules through the center of the pore. So a lot of the structural work in this area has been done in mammalian and in yeast cells, but there have been a few studies in plants as well. A real seminal study was done about 10 years ago now, looking at tobacco BY2 cells and looking at the outside of the nucleus using scanning electron microscopy. And you can see here, as you look across the outside of the nucleus, you can see these structures which are reminiscent of nuclear pore complexes from other systems. And if you look in a little more detail, you can see that nuclear pore complexes are shown in eightfold symmetry around this central pore. And each of these individual blobs basically hold a whole load of different proteins which make up one component of the nuclear pore complex. And then this whole thing is repeated 8 times around. So we look in a little more detail. So each of those blobs, as I said, is made up of a lot of individual subcomplexes. And then within those subcomplexes, there are individual proteins. And oftentimes these are called nuclear porins, or abbreviated to NUP. You can see here, this highlights the number of nuclear pore proteins, nuclear porins, which are present in the plant. And for the most part, plant systems share the same number of nuclear porins with mammalian and in yeast systems. However, there are a number which are found in mammals or in yeast, which have been crossed out in this diagram, which show that these actually don't exist in plants. And the converse is true, that there have been a couple of nuclear pore proteins that have been found in plants that don't have any homologs in mammalian or yeast systems. And therefore, so although the overall idea and the overall function is the same, there is some specificity between these systems. So of course, as I said, a lot of this work has been done in single-cell yeast or in mammalian cell cultures. So the power of plants, of course, is you've got a multicellular organism, and you can do genetics on it to see what the consequences is of knocking out individual members. So what we can do in plants, and particularly in Arabidopsis, we can identify plants which have a mutation in different nuclear pore proteins and then see what happens to the plant. So oftentimes, surprisingly little actually. So a number of mutants which have a mutation in nuclear pore proteins really look very much like the wild type plant. So in this case, it's the wild type plant on the end, and then these two mutants, they look very much the same. Whereas there is a classic sort of nuclear porin phenotype that you get, where the plants are smaller, they flower earlier, they have a smaller root as well, they flower earlier, and they're less fertile. And this is what we see in this NUP96 and this NUP160 plants. Another more molecular phenotype that we see in these is interesting, is that we see a defect in the transport of messenger RNA out of the nucleus into the cytoplasm. So what we see labeled in these leaf cells from the wild type or in the nuclear pore mutant, this is labeling of the entire of the total messenger RNA within the cell. And you can see in these certain nuclear pore mutants, you can see that this is held within the nucleus. So therefore, the function of the nuclear pore complex in this case is to help transport messenger RNA out of the nucleus. We see that this happens. The function of this is, well, we understand the function, but the consequence of this is really not known at the moment. This is under continued study. So what we showed, or I just showed, is that a lot of these nuclear pore mutants have relatively mild phenotypes, but we do see things which are much more severe phenotypes. So if you knock out a number of other of these nuclear pore components in plants, the plant is unable to survive. And this is kind of what you'd expect. So something as fundamental to the plant as the nuclear pore complex, this transport between nucleus and cytoplasm, if you remove a number of those components, the plant's unable to survive. And maybe, as I said, this isn't surprising. It's what maybe is more interesting is what we see in those other mutants where the plant is able to survive. And we really want to understand where the specificity is and why that is able to happen. So what about the nuclear pore complex and actual gene expression within the nucleus? So this is a classic picture which comes from a mammalian cell line, and you see this portion is within the nucleus and this section is the cytoplasm. And you see the darker stained regions. This is heterochromatin, so inactive chromatin, which you can see goes to the edge of the nuclear envelope. And then where the red arrows are, this is where the nuclear pore complex is. And you can see quite clearly that this inactive chromatin is around the outside of the nucleus, but in the vicinity of the nuclear pore complex, where this nuclear basket structure was, you can see that there's no heterochromatin, and this is a more open area. And this goes along with a theory called the gene gating theory, in that messenger RNAs travel towards the nuclear pore complex and they're able to preferentially move through the pore complex in order to be rapidly translated. So we don't have a huge amount of evidence for this in plants, but there is some evidence that there's differences in expression of loci which sit around the periphery of the nucleus, and also some work from Oxford Brookes University where they've tethered different proteins or reporter proteins to the nuclear pore complex, and they show that their expression is increased. We also know that this nuclear basket within the nuclear pore complex interacts with the Saga complex and the TREX2 complex, which are involved with taking messenger RNAs and transporting them towards the periphery of the nucleus. So it does appear that the nuclear pore complex plays a more active role in the control of gene expression. So what we know from this system is that we have heterochromatin exclusion surrounding the nuclear pore complex. There is the association with active genes, and there's also direct chromatin binding of nuclear pore proteins when they're away from the nuclear pore complex. So it may be possible that nuclear pore proteins bind to chromatin, and then they bring those loci towards the nuclear pore complex when the nuclear pore complex is reformed. And there really has some work to be done in this area. So all the papers that I mentioned, all the research I mentioned in this talk, they're in the notes for this YouTube video. You can find all the references there. So if you want to continue on with any of this of your learning in this area. So for the In-Depth Academy, we like to end with 3 questions. So firstly, how does the nuclear pore complex regulate the transport of specific molecules? So this is the big question. Do the nuclear pore complex proteins specifically interact with transport components, or is there a more general effect? How is the movement of loci within the nucleus to the nuclear pore complex regulated? So we don't really have a good idea for this. I mentioned a few ideas, but this is something that we need to understand a lot more about. And then a bigger question is, how is the composition of the nuclear pore complex? How does it change, or is it the same in all cells? So if we have more quiescent cells, so large leaves that aren't growing active, large leaf cells, sorry, that aren't actively growing, do they have a different composition of the nuclear pore complex than those meristematic cells, which are growing more actively? Is it the same composition, or are there just less of them? And this is something we we don't understand yet within the plant nuclear pore complex, and that's something we'd like to look at for the future. Okay, so I hope you've learned something today from the INDEPTH Academy. If you have any questions about this, please don't hesitate to contact me, and my contact information is on the within this YouTube video as well. So thank you much for listening.